Welcome to day 5 of distance learning. Hopefully you've had a good, week, a good day. We're going to start talking about mutations today. And so some of this will be a review, but some of it will be new, and I'll get to that in a minute. So after today's lesson, you should be able to know the difference between a chromosomal mutation and a gene mutation. Gene mutations we talked about before, and I'll remind you of those. Identify at least 2 types of chromosomal mutations and identify at least 2 types of gene mutations as well. So what are mutations? Any type of change to the DNA, right? Any type of change to how those A, T, C's, G's are arranged. This can occur in somatic cells. Remember, somatic is the fancy way of saying any cell in your body that is not a sex cell. So it's not sperm, it's not egg. It could be a liver cell, heart cell, eye cell, cheek cell, any of those. And those don't get passed down to offspring. They can also occur in gametes, and we talked about that earlier in this unit when we talked about non-disjunction and meiosis. I'm going to briefly touch on that again today as well. So are they good? Are they bad? Well, I think I kind of addressed that in the other animated video that I made. But again, mutations happen regularly. So the majority of mutations are just neutral, meaning they're not really good or bad. They're just there. Chemical exposure, UV radiation, any of those things can cause mutations. That's why skin cancer, that's a type of mutation. If you think about that, that's from the UV radiation from sun. And most of them get fixed by enzymes, and we're not going to get into that in detail. So really, again, are they helpful or harmful? Really, we'll talk about that in the evolution unit. It kind of just depends on your environment. Some types of skin cancers come from those somatic mutations, again, of those body cells. And sometimes these mutations, again, do help an animal's chance for survival. So we'll talk about that a little bit later. So let's look at types of mutations. So a chromosomal mutation, that's when we're looking at the entire chromosome structure, okay? And so we're actually changing the whole structure of the chromosome. That means it's a lot of DNA because there's a bunch of DNA that's coiled, and that's what makes a part of a chromosome. This can be a loss or a gain of a chromosome as well. So 5 different types that we have: deletions, which I'm sure you can kind of guess what that is, inversions, translocation, non-disjunction, which we talked about with meiosis, and duplication. And I'm sure that duplication and deletion kind of, you can guess those on the words, and I'll talk about the other 3 in a minute. So deletion, again, part of the chromosome breaks off, so it gets kind of lost. So I showed you in this picture, what we have here is, let me kind of get my pen over here. This whole piece in this chromosome, gone. So eventually we end up with this. Where did this information go? We don't really know. And I'll talk about that in a minute as well. An inversion is when the chromosome segment breaks off and it inverts itself. It flips around. So it's a little bit backwards. So let's pretend again we had, here's our, I'll use the same pen color. So here we have this part of the chromosome. It breaks, it flips itself, and now for some reason the green part is on the top, the red part's on the bottom. So the information's still there, but it's inverted, which is not necessarily a good thing. Okay, duplication, we get a segment of the chromosome that gets duplicated. So you can see instead of 1 yellow section, we have 2, it gets repeated. And with translocation, this is kind of similar to inversion, but a little bit different because it's 2 chromosomes. The information, I'm going to hide this, the information breaks off. Okay, so you can kind of see here, the information breaks off and it attaches to a completely different chromosome. So the green that was on chromosome 20 got attached to chromosome number 4. Okay, the blue that was on 4 got attached to 20, completely different. Okay, they just kind of went the other way. All right, again, just giving you that, how that kind of looks. Just using the example of chromosome 4 and 20. It can happen with any chromosome. All right, non-disjunction, this should be a review. We talked about it. It's the failure of chromosomes to separate during meiosis. Remember, they kind of stick together. And so we get gametes that either have too much genetic information or too little genetic information, which we don't want. Okay, and this was just kind of a highlight. So if that happens, again, we can see that kind of occurring here. These 2 got stuck together. Oh no, this has too few, this has too much. Over here, we end up producing gametes, too much DNA, too little DNA. Not a good thing. This is just giving you that animation. So take a minute just to kind of watch. That was a deletion. This is an inversion, that flip. This is a translocation where we've switched parts of 2 different chromosomes. That's a duplication. And then that's at the original. Now remember, these letters are just meant to represent nucleotides or parts of the chromosome themselves. All right, again, just kind of giving you that image on what that can look like. I just put that there, so if you want another picture, you can look at another picture. Okay, let's get into gene mutations. This should be a review for you. So this is when a nucleotide sequence of a gene is changing. This is when the A, T, C's, and G's of DNA are switching around, okay? It's usually, again, gonna involve a single nucleotide. So point mutations, substitutions, insertions, deletions, and frame shifts. We talked about those earlier. I'm just gonna do a quick review because that might have been new. So again, a point mutation is one single nucleotide, one A, one T, one C, one G changing, okay? And so in this case, you can see this is what the sequence should have been if we had no mutation. There's different types of point mutations. Again, remember, not all mutations are bad, not all of them are good. The majority of them are neutral, meaning they're not helpful or harmful. So in a silent mutation, that's when you have a mutation happen. So instead of C, I got T, but guess what? I got the same protein made. So I wouldn't even notice that mutation occurred. A nonsense is when we get an early stop signal, right? So that T became an A, it caused UAG to happen, it's a stop. Missense means I'm just getting a completely different protein that's being made, right? So in this case, I'm getting ARG and THR. All right, so a real world example of this would be something called sickle cell anemia or sickle cell disease. And this is where your blood cells should look like this. So we should have these blood cells that look almost like jelly-filled donuts. But with sickle cell disease, we see these blood cells, these red blood cells that looked curved and they're not very full and they kind of stick to each other and they fold and they're irregularly shaped. And so what that happens, what causes that is a mutation. Now, what's the effect of that? It's actually just poor oxygen delivery in your body. And I'll talk about that a little bit as well today. And you'll look at that a little bit later with the lesson. So what happens? So this is what we do want. We should have CTT, which would make GAA, which gets us this protein, which is glue. What happens in the case of sickle cell disease is instead of having that first T, we get an A, we get adenine. And so that produces a completely different protein. And that different protein is what causes those red blood cells not to be shaped properly, okay? And we'll talk about that later. I just wanted to give you that's just one thing that can have a huge outcome. All right, a frame shift mutation is going to completely move the reading frame, okay? So we're either gonna insert a base or delete a base, and that's gonna shift how we're reading. So I kind of gave you a little sentence to help this a little bit more. So the original should be the fat cat ate the wee rat, right? If we were to add an A, it shifts how we read the rest of the sentence. The fat cat at. So that moved how we read the sequence. Remember, we read a codon is 3 of those bases, right? A, T, C, or G. It's 3 of those. So we have to read 3 at a time. So it shifts everything down. Okay. Again, this is a frame shift mutation as well because we deleted that G that you see there. Okay, here's just an animation. This is a substitution where we just put one in for another. That's an insertion we added, deletion we take away, and inversion we flip them around. Okay, so I'm hoping that the chromosomal mutations were new. Again, just kind of looking back at what our goals were for today. The chromosomal mutations were new, but the gene mutations hopefully were a review. And that's it. Have a lovely rest of your day.